The adult brain undergoes continuous structural remodeling. Neural stem cells actively divide, differentiate, and extend their dendrites to integrate into existing active circuitry. This structural remodeling is localized to two specific regions: the subventricular zone, or SVZ, in the wall of the lateral ventricle, and the subgranular zone, or SGZ, located within the dentate gyrus of the hippocampus. These newly generated granule cells must functionally integrate into the trisynaptic hippocampal circuits. The trisynaptic circuit is a relay of three distinct synaptic connections, from the entorhinal cortex to the dentate gyrus, then to the CA3 and CA1 regions, which modulates spatial memory and mood. This process of cellular integration is sensitive to physiological states. Pregnancy and the postpartum period represent an extreme physiological environment, defined by systemic endocrine fluctuations that alter the brain's architecture. This chart tracks estrogen, progesterone, and prolactin across a 40-week gestation period. All three increase steadily up to parturition, or childbirth. Following delivery, estrogen and progesterone experience a cliff drop to baseline. In contrast, prolactin remains elevated to support lactation. This volatile hormonal environment dictates the survival and proliferation of neural stem cells. These chemical swings provide the biological mechanism driving the cognitive adaptations required for motherhood. Establishing the functional significance of these new neurons requires precise experimental ablation. By eliminating them, researchers can observe subsequent behavioral deficits. Standard methodologies like fractionated X-ray irradiation or infusions of antimitotic drugs like Ara-C lack cellular specificity. An antimitotic is a substance that prevents mitosis or cell division. Because these drugs circulate systemically, they create widespread physiological noise. Genetic ablation models such as Nestin TK transgenic mice also present challenges. These models use genetic triggers to kill specific cells, but the process can induce off-target physiological stress that interferes with behavioral data. To bypass these systemic side effects, researchers utilize iodine-125 seed brachytherapy. This technique involves the focal placement of a radioactive seed directly within the dentate gyrus. This localized radiation zone specifically eliminates actively dividing neonatal neurons. It spares the surrounding tissue and complex cytoarchitecture, preventing generalized brain trauma. This chart maps the cellular timeline verified by this methodology. It tracks progression from a neural precursor cell, marked by BDU or Ki67, to an immature neuron identified by DCX, and finally to a mature neuron expressing the new N biomarker. This level of spatial precision allows researchers to isolate the behavioral outcomes of neurogenesis in the subgranular zone, separated from the variables of systemic trauma. With clear methods established, the data reveals a neuroendocrine paradox. High levels of pregnancy hormones do not produce a simple, linear increase in new neurons. This matrix compiles data from adult OVX mammalian models, subjects where the ovaries have been removed to control baseline hormone levels. The table correlates various estrogen species and exposure times against subsequent neuron proliferation. The data for acute exposure is consistent. When subjects are exposed to estradiol or estrone for under 4 hours, the dentate gyrus exhibits a significant upregulation in cell proliferation. Chronic exposure produces the opposite effect. If estrogen exposure is maintained for 20 to 28 days, it results in neutral or downregulated proliferation. This chronic suppression occurs because sustained estrogen stimulates the HPA axis. The HPA axis is the hypothalamic pituitary adrenal system that coordinates the body's stress response. Its prolonged activation releases cortisol, a hormone that actively suppresses cell division. The primary risk to the brain occurs during the sharp decline of these hormones after delivery, known as postpartum estrogen withdrawal. This rapid withdrawal triggers massive apoptosis, or programmed cell death, in immature progenitor cells. The sudden lack of estrogen dismantles the new cellular architecture. This localized structural degradation in the hippocampus correlates with negative behavioral phenotypes. Animals subjected to estrogen withdrawal show steep increases in markers for postpartum depression and anxiety. Estrogen acts as a temporal trigger. Short-term spikes build the network, chronic exposure stalls it, and abrupt withdrawal destroys the architecture. Progesterone operates alongside estrogen, functioning specifically to enhance the early-stage survival of newly born neurons. This visualization compares the hormonal effects. On the left, progesterone alone causes a mild upward trend in cell proliferation. On the right, we see the effect of progesterone administered concurrently with estradiol benzoate, a synthetic form of estradiol used for precise dosing. When combined with high-dose estradiol, progesterone acts as an antagonist. It effectively blunts the acute proliferation peaks induced by estradiol alone, regulating the pace of neurogenic growth. While estrogen and progesterone regulate the building process, prolactin serves a different function. This pleiotropic hormone, one that produces multiple distinct biological effects, is secreted during the third trimester and throughout lactation. Prolactin binds directly to specific receptors localized within the subventricular zone and the hippocampal dentate gyrus. By binding to these receptors, prolactin counteracts glucocorticoid damage. It functions as a neuroprotective shield for dividing precursor cells during postpartum stress. This shield is a biological necessity. Mice engineered without prolactin receptors show impaired hippocampal-dependent learning, confirming that prolactin is required to maintain cell division under stress. The system relies on hormonal synergy. Estrogen initiates construction, progesterone modulates the pace, and prolactin shields the delicate circuitry from the high-stress endocrinology of the postpartum period. The dentate gyrus functions as a volatile physical architecture, its structure shifting in response to hormonal timing. The maternal brain operates under a tripartite vulnerability, depending on acute estrogen for proliferation, relying on prolactin for stress protection, and threatened by rapid endocrine withdrawal following delivery. Mapping these specific cognitive effects requires highly localized methodologies. Systemic models create too much interference. Only precision tools like iodine-125 brachytherapy can isolate the behavioral outcomes of hippocampal neurogenesis. This cellular plasticity ties to the clinical realities of motherhood. The growth and survival of these neural circuits dictate a mother's spatial memory and cognitive adaptation to her offspring. The rapid loss of these neurons during hormone withdrawal compromises hippocampal circuits responsible for emotional stability. This suggests that postpartum mood disorders stem from a physical degradation of neurogenic architecture. Recognizing this cellular framework allows for the development of targeted neuroendocrine therapies.